to talk on decode computational and metabolic insights into no bacterial mediated management for rice PLB. Thank you. You have 10 minutes, Roy.
Good afternoon to everybody present for this session. Today I'm going to talk about the computational and metabolic insights into the actinobacteria mediated management of bacterial leaf blight of rice. Rice feeds half the million population of the world, and a major hindrance to the rice production in this world is caused by bacterial leaf blight of rice, which is caused by Xanthomonas oryzae, Petavar oryzae. And you can see that chloropleth map in which India comes under a high risk zone for bacterial leaf blight susceptibility. As the world is shifting towards sustainable management, there is a high need in the hour for the biological control management of rice. And why actinobacteria? Because they are a very good plant growth promoting agents, and also they are elicitors of plant defense, and also they are a very good biological control agent. Isolation and enumeration of the actinobacteria from rice rhizosphere and the screening of these actinobacteria against the Xanthomonas oryzae, Petavar oryzae. Potent actinobacteria, which are screened through the primary and secondary screening, were further tested in glass house and field condition. Metabolomics profiling of the potent actinobacteria using the HRLCMS method, and the molecular interaction of the pathogenicity factors of SU with the actinobacterial metabolites in silico conditions under molecular dynamic modeling. This is the flow of the study. These are the diverse actinobacterial isolates from the rice rhizospheres, isolated using the serial dilution method in starch casein agar medium. And primary screening of the actinobacterial isolates was done using the dual culture plate, and the 8 isolates were found to be effectively reducing the disease suppression, and they were taken for secondary screening in disk diffusion assay, and AMGD 1620 and AUDT 502 were used to were very effective to control the bacterial leaf blight of rice. And molecular characterization of the AMGD 16 was found to be Streptomyces gingivanus, and AMGD 20 was Streptomyces xiamensis. And when we're biochemically characterized, they were producing sidrophore. They were positive for IAA production, catalase production, and also HCN production and phosphorus solubilization. And when taken under field evaluation, the seed treatment at 10 liter per kg and 2 foliar spray of 10 ml per liter with AMGD 16 at 10 raised to 8 CFU per ml had enhanced the root length, the plant height, reduced the lesion length, and which showed that they are good plant promoting and disease suppression agents. The AMGD 16, which was a very good biological control agent, were further taken for HRLCMS analysis to understand its metabolome. And based on the area of abundance, different metabolites were selected, and literature mining was done to understand the function of those metabolomics, and we saw that there are different indole derivatives like indole 3 acetic acid, indole 3 carbinol, methyl indole 3 acetate, and also indole 3 acrylic acids and pyroxidine. They are all different very good growth promoting or metabolites, and also lumichrome, fusidic acid, and manumycin, oleomycin. They are all different antimicrobial agents which can be used for further reducing the Xanthomonas disease suppression of bacterial leaf blight. This is the total ion chromatogram of the actinobacterial isolate plotted by relative abundance against the time retention time. Different peaks show the relative abundance of different metabolites, and several peaks show the diverse nature of metabolites produced by the AMGD 16. And in order to understand where each metabolites belong to which pathway, the global pathway enrichment analysis is done, and each bubble represented the different pathways. And the major pathways were found as valine leucine, isoleucine biosynthesis and degradation pathway, the pyrimidine metabolism, purine metabolism, tryptophan metabolism. They all have different roles in plant growth promotion and also microbial suppression. And further, to understand the molecular level of interaction, two pathogenicity factors, that is PTHO1 and peptide deformylase, which are very important for the virulence and the viability of the zoo, were taken, and the molecular docking studies was conducted between the different metabolites and also the pathogenicity factor. And PTHO1 binds with the promoter region of OH sweet gene 11, which will upregulate and lead to the sweet sugar influx. And this is the Ramachandran plot showing the backbone dihedral angle distribution between the phi and psi angles of the amino acid bases. And most of the amino acids were in the allowed and disallowed, in the favored and the allowed regions. And you can see that lumicrome has bound with a minus 6.6 kilocalorie, binding energy of minus 6.6 kilocalorie per mole, and this had the least binding energy, and this was and had three number of hydrogen bonds, which was further taken for dynamic modeling to understand the stability of the protein. The RMSD plot also shows that in the initial phases there was fluctuation, but towards the 100 nanoseconds the entire complex was stable. And when we had seen the RMSF, that is the root mean square fluctuation, only controlled fluctuation was there, and the radius of gyration also showed that the entire protein structure was compact, and the more number of hydrogen bond was found throughout the entire over time, which shows the stable nature of the complex. And the superimposed structure also shows that it is an induced fit model, which shows the stability of the protein and the ligand. And further, PTHO, the peptidyl deformylase protein also was taken, and here also the stability, the stereochemically stable protein was present. And here also lumicrome had bound with the peptidyl deformylase with a binding energy of minus 8.3 kilocalorie per mole. And here further you can see that the RMSD plot, fluctuation, RMSF plot, and all the plots show the stabilization of the protein. And the superimposition showed a induced fit model. The key outcomes are AMGD16 as seed treatment and 2 foliar spray reduce the bacterial leaf blight incidence to 32.17%, and the lumicrome effectively bound with the pathogenicity factors of the xanthomonas. And lumicrome can be considered for the future applications can be lumicrome can be suitably a commercial scale production can be undergone through microbial fermentation, and it has both antimicrobial and as well as a growth promoting character. And till now, no direct antimicrobial agent like lumicrome was never used as an antimicrobial agent in plant disease management. So this is one of the studies where the importance of lumicrome is well documented, and the structural stability is being shown through dynamic modeling and molecular docking. Thank you.
Thank you, Ms. Ashita Roy, for an excellent overview and also sharing some insights on actinobacteria-mediated management of rice BLB. Thank you so much. Any quick questions to Ms. Ashita Roy? Thank you. I think last 3 hours we talked about and we heard a lot of story or findings on the rice. Now we have 2 next presentations which are on, one is on brinjal and other one on sorghum. So first I would like to invite Mr. Nikhil Kumar to talk about decoding the metabolites during the fruit development for disease tolerance in brinjal.
Good afternoon, and one and all present here. I am happy to present one of the interested crop other than the major crops. Here I will be presenting our work decoding the metabolites during the fruit development for disease resistance in brinjal, Solanum melongena. Basically, eggplant is a widely cultivated and economically significant crop in the Asia, and like in India also it is like major crop used as a food. and it is susceptible to fungal, bacterial, insect pests, and it leads to yield loss and reduced marketability. The secondary metabolites like phenolics, flavonoids, anthocyanins, glycoalkaloids not only contribute to taste, but also it plays an active role in plant defense against biotic stress. Defense responses like secondary metabolite accumulation depend on developmental stage and tissue type. This untargeted metabolite captures the global changes in defensive metabolites, which helps to identify the biochemical markers and pathways involved in fruit defense and disease resistance. This is the one special variety called Solanum melongena variety Mattugula, which is only grown in Mattu village of the Udupi district, which bagged the GI tag in 2011 from Chennai. For this experiment, we have selected 3 fields in which we have selected 25 plants. In each plant, we labeled 5 flowers, and later, in the time interval of 15 days, we have collected the samples. Later, the peel and pulp were separated and metabolite extraction were performed using acidified methanol, followed by homogenization, sonication, and concentration. This table 1 represents the gradient program for LCMS and the instrument parameters, and this figure 4 represents the workflow which has been performed in the Compound Discover software. This figure 5 represents the multivariate analysis for the 15 dpa peel and pulp samples, where we have found 400 plus metabolites, in which we have found 123 common metabolites, where 44 metabolites were upregulating and 47 metabolites were the downregulating. Whereas in 30 dpa we have found that 330 metabolites, where 77 metabolites were found in both peel and pulp, where 10 metabolites were upregulating and 19 metabolites were downregulating. In 45 dpa samples, whereas the pulp sample we have found that more metabolites, and for only 45 metabolites we have found a common in both. When compared to the initial stage of the development to the final mature stage of the development, the common metabolites are reducing and the unique metabolites were increasing. So we thought to check what are the major disease resistant metabolites and the metabolites related to defense. So we performed that analysis, and we have found that here we have compared in between the stages. Previously we performed the analysis among the peel and pulp of the same stage. Here we performed in between stages. This slide represents the disease resistance metabolites in peel samples, among 15 versus 30 dpa, 30 versus 45, and 15 versus 45. We have found that only few metabolites which are found common in both stages. Other than that, there are the many unique metabolites. This is the disease resistant metabolites in pulp sample. Here, when among the development, the number of resistant metabolites were increasing when compared to the early developmental stages. And later we performed the metabolite pathway analysis, where we have found that the compounds which we have identified for disease resistance, they were undergoing pathways like phenylpropanide and flavonoid pathway, oxylipine and fatty acid signaling, amino acid defense pathway, steroid alkaloid and cyanogenic glycoside pathway, and terpenoid metabolism. To conclude, in this study, a wide and targeted metabolomic approach was performed to identify disease-resistant metabolites. A total of 1,041 metabolites identified in 3 stages, among which more than 45% of the metabolites were identified as disease-resistant metabolites. This fruit development involves the progressive metabolic reprogramming, where peel tissues accumulate defense-related metabolites in stage-dependent manner, forming a chemical barrier against the disease. These tissue-specific metabolite profiles suggest active metabolism, likely reflecting differential roles of peel and pulp in defense, transport, primary and secondary metabolic process during fruit development. These are the key references used for this study. And I would like to thank my mentor, Dr. A. Muthuswamy, for his constant guidance and support. I would like to thank Manipal Academy of Higher Education, Manipal School of Life Sciences for their infrastructure and facilities. And I would like to thank SERB and ICMR for funding to our lab. And this is our team working on the broad aspects of the plant biology. Thank you.
Thank you, Mr. Nikhil, for sharing some of your work on metabolic pathways and also profile. Thank you for the excellent work and sharing with all of us. Any question? Yeah, please, sir.
Thank you. Good work.
Thank you, sir.
There is one species called Solanum macrocarpon.
Yes, sir.
It is called tomato brinjal and also edible. The good part
yes.
is it is edible.
Yes, sir.
And if not immune, it is very highly resistant.
Yes, sir.
I would request you to parallelly compare the
Sure, sir.
metabolites with the macrocarpon with melangina
Sure,
and
sir.
see where exactly we can gain.
Sure, sir. We'll carry on.
Okay, no more question. Thank you again.
Thank you, sir. Thank you.
So I'll invite now the last presenter for the today session, Ms. Ranjita, to talk on identification of marker trait association for fall arboretum in resistance in case of sorghum.
Good afternoon, everyone. I will be presenting on the topic identification of marker trait association for fall arboretum resistance in sorghum. This study focuses on identification of the genome-wide SNPs and their association with fall arboretum resistance. This pest is recently invaded across the globe. So, fall arboretum, Spodoptera frugipeda, is native to America. In 2016, it was first reported outside its native in Western Africa. Then in 2018, in India, at Karnataka. So, till now, it has invaded more than 80 countries across the globe. With this rapid dispersal ability, it has become a polyphagous pest, infesting more than 80 crops, host crops, majorly maize, sorghum, and rice. And we know that sorghum plays an important role in food and fodder system, especially in semi-arid tropics regions across Asia and Africa. So this pest is causing yield losses up to 30 to 80%, and currently chemical control it is in management, but it is ineffective as fall arboretum feeds inside the rolls, and this pest also has developed a resistance to many of the insecticides. So host plant resistance is a best alternative approach. So with this background, we have framed our objective is to identify the molecular basis of resistance to this pest. We have taken 242 minicore sorghum accession from ICRISAT, and this minicore panel represents the global diversity. It helps in identification of rare alleles as well as novel genes. So this is our, we framed in multi-step screening, including a field as well as laboratory. So in field we have screened under natural infestation condition as well as in artificial infestation condition using a screenhouse facility available at ICRISAT for the across 2 seasons. Here the observations we have taken are like foliar damage score, glossiness, vigor, and leaf sheath pigmentation from the scale 1 to 9 Davis scale for the foliar damage score, and other traits 1 to 5 scale, and under artificial also foliar damage scale for 1 to 9 scale. And to take the account of antibiosis mechanism, we have also conducted detached leaf bioassay for the same panel 242 mini core collection, and we have here we have taken leaf sample and kept in agar medium, and we have infested the 10 neonate larvae, and after 5 days we have taken observation for the leaf damage score, then number of larvae survived, and the weight of the survived larvae. So from this we have taken bluff values, and we taken further the genome-wide association study. That is all about phenotyping, and genotyping. We have GBS data already available at ICRISAT. So we have aligned that GBS data to sorghum reference genome, and we have got 1,73,000 SNPs. And after filtering the minor allele frequency, we have got 1,70,000 high quality SNPs. So after that, we have corrected with the population structure. Then did GWAS at using GAPIT package in R, using a multilocus models like Blink, Farm CPU, MLMM. At a significant threshold of 2.9 into 10 to the power of minus 7. So after that, the resulted SNPs we have parsed with this GIF annotation file taken from NCBI version 3.62. Then after that, we have checked for genes present within plus or minus 50 kb LD region. So coming to the results, all the traits showed significant genotypic variation with more than 0.8 heritability for leaf sheath pigmentation and moderate heritability for glossiness and others traits with low heritability. And when coming to the correlation between the traits, the trait vigor, glossiness and foliar damage score are they are moderately associated, but here leaf sheath pigmentation we got weak correlation, but earlier studies showed that leaf sheath pigmentation is contributing resistance to pest, and recently in sorghum also the flavonoids are shown detrimental to the fall armyworm. So as they belong to ordinal data, so we did correspondence analysis where we converted the scores into resistant classes for both the trait leaf damage rating and leaf sheath pigmentation. So after association, we got certain classes like partial resistance and susceptible classes were strong. They showed a strong association. So after the GWAS, we have got a total 45 significant marker trait association for the 4 traits: foliar damage score, leaf damage rating, leaf sheath pigmentation, and larval weight. And some of the traits you can see leaf sheath pigmentation and larval weight as well as LDR showed more. They occurred in more than one models. So these are the total 45 significant MTAs. And through Blink model, we have got 10 significant MTAs where you can see the p-value ranging from 10 to the power minus 7 to 10 to the power minus 100. And it also showed good phenotypic, they explained a good phenotypic variance ranging from 3 to 66% of the variance. And so you can see the Manhattan and respective QQ plots of the association. The foliar damage score 1 for 1 SNP and 4 significant SNP for leaf sheath pigmentation, and 2 are on the same 6th chromosome, and 1 for LDR, and 4 for survived larva weight, where 2 are on the 1st chromosome. So after the annotation, we got know the genes, candidate genes present within plus or minus 50 kb, where we got this description of the identified candidate genes. Example, FDB for the trait LDR, and then other proteins for the leaf sheath pigmentation like kinase, and as well as CBS domain. And then this larval weight actually showed a good related proteins like proline dehydrogenase and pectin acetylase, and as well as peroxisomal nicotinamide. And also for LOH2 related to lipids and related to calcium flux and glycosyl transferase, as well as serine or threonine specific proteins. So previous studies also have reported some of the proteins like peroxisomal nicotinamide. They play important role in jasminic acid, other oxidative stress related pathways, actually through flavonoid based host defense. and calcium binding family proteins. The calcium flux will help in signaling. And also the other proteins like pectin acetylase and lipid related LOH2, and they are involved in cell wall remodeling, and other things like glycosyl transferase involved in salicylic acid mediated signaling. So in conclusion, our study GWAS on 242 sorghum mini-core collection, we have identified 45 significant marker trait associations for the fall armyworm resistance, and through blink 10 MTAs for the 4 trace, explaining 3 to 66% of the phenotypic variance, supporting the involvement of multiple loci from small to large effects. And annotations showed that they are involved, those candidate genes are involved in some signaling, redox regulation, flavonoid biosynthesis, as well as cell wall modification. So in future, the validation of the identified candidate genes are needed, then using of transcriptomics and metabolomics to understand the mechanism of resistance. And then next, the development of markers for these candidates, and next further involvement of them in marker-assisted breeding. Thank you.
Thank you, Ms. Ranjita, for sharing outstanding work, and also to demonstration of some of the tools what our earlier presenters presented, both in analytics and also molecular tools. Thank you for that overall. And now I request Dr. T.S. Sharma to share some conclusion.
Thank you, Dr. Gowda. So in this particular session, we had 8 presentations, including 3 oral presentations by our young colleagues. And you all will agree with me that this was one of the most enriching and knowledgeable session which we had on rice. And I would like to say 4 to 5 points on this particular sessions. Particularly, we learned about disease and insect pest scenario of rice in the country, their prevalence, significance, and management practices. Then a success story of discovery and deployment of blast resistance gene PI54 and its impact on rice breeding. And then mapping and characterization of genes for various biotic stresses and their introgression in rice breeding using marker-assisted selection, and also successful examples of mass-derived products which we have in the country now, and then challenges and opportunities of rice improvement globally, and available new strategies with the breeders. And finally, we had 3 presentations by our young colleagues showing their work, and I would like to congratulate all the speakers, particularly our young speakers, for their nice presentation. And I would also like to thank my co-chair, Dr. Viresh Gowda, head R&D Wire Biosciences, and 2 reporter, Dr. Suma and also Dr. Gurudatta. Besides that, thanks are to the organizers for nicely conducting these particular sessions and giving us opportunity to be here this morning. Thank you very much.
Thank you so much, sir. Now we take the privilege to extend our sincere gratitude to our learned speakers. I call upon Dr. M.S. Prasad, sir, Dr. R.M. Sundaram, sir, and Dr. Amresh Chandal, sir. Please grace the dais, and I request our Honorable Vice Chancellor, Dr. P.L. Patil, sir, to do the honors. And I request chair and co-chair also to join the felicitation of our learned speakers. Yeah, now further we take the pleasure of felicitating our respected chair and co-chair also on behalf of University of Agriculture Sciences, Dharwad. So to Dr. Videsh Gowda, sir, who is a proud alumnus of this prestigious University of Agriculture Sciences, Dharwad, who did his masters in this university. And to chair now Dr. T.R. Sharma, sir, former Deputy Director General, ICAR, New Delhi. Thank you, sir. And we also extend our sincere thanks to both the rapporteurs, Dr. Suma Biradar Madam and Dr. Gurudat Hegde, sir. And we request them to hand over the proceeding of the session by tomorrow morning. Thank you, sir. Now we have a very special event of exchange of MOU between University of Agricultural Sciences, Dharwad, and a private Maize company. I request Director of Research, US Dharwad, to join us on the stage. And also the representative from the company. And I request Honorable Vice Chancellor, sir, to speak few words on this occasion.
Yeah, thank you, Dr. M.D. Patil. So you can take this is a commercial break or a commercial opportunity. So we want to commercialize our technologies released by our own scientists, maybe varieties, hybrids, and also the important inputs. So kindly refer to, I request all the industry partners, please refer to Agri Innovate India Limited website. We are commercializing all our technologies through Agri Innovate India Limited, and choose the technology required and express your interest, and through the material transfer agreement, we will have the commercialization of our technologies. And we have already commercialized 2 color cotton hybrids, maize hybrid, and biofertilizers. So like this, the list is go on. And also we have several the value-added products from College of Community Science. They are also available for commercialization for the young entrepreneurs or ladies to take up the activities. So here, today, so we are having MOU between one industry, which is that, the seed company?
Sir, this is
Gen Z Seeds and US Dharwad for our Dharma hybrid and another the line in the another hybrid in the pipeline. So Dharma hybrid, the features will be indicated by our maize scientists. So both are US Dharwad products. Each are sold for 3.6 lakhs, I think 3.3 or 3.6 lakhs. So in addition to Agri Innovate India, so you can also consult Directorate of Research US Dharwad for commercialization. Thank you very much.
just to brief you the variety details, and with the support of our Vice Chancellor and successful licensing of our PPMC head Dr. C.R. Patil sir, we organized one maize hybrid field day during September. There around 15 private partners participated, and they shown the interest in some of the hybrids. And this is a one fruit we are getting out of that one. And there are so many people hybrid they are asking for. to evaluate in their field, and they may come. They have shown interest in next course of season. They will come and to have MOU so that the fruit of our scientist will go to end user, that is a farmer. These are the effort of our two breeders, Dr. S.C. Talekar and Dr. Kachapur, sir.
To move further, now we have a panel discussion on this theme of on-the-crop rice. I request Dr. R.M. Sundaram, sir, Director ICR IRR Hyderabad, to chair this panel discussion. And then I call upon Dr. N.G. Ramachandra, Lead Breeder, Rice Breeding Bioseeds Hyderabad. Dr. Venkatesh Kulkarni, Director R&D, Nath Biogene Seeds. Mr. Devaraja Achar, Head Biotechnology, Indo-American Hybrid Seeds, India Private Limited, Bengaluru. Dr. Chandrasekhar S., Rice India Breeding Lead, Koteva, Hyderabad. Dr. S.K. Prashanti, Professor and Head Biotechnology, US Dharwad. And Dr. Viresh Gowda, Head Asia Rice Breeding, Bayer Crop Sciences, Hyderabad, to join as a panelists.
Yes, sir. Yes, sir, you can come.
Okay, that'll be fine. I look the odd man.
Good morning to you all, rather, good afternoon to you all. We are here for an important panel discussion on the theme, particularly breeding crop varieties for durable resistance to multiple biotic stresses, how we have progressed from genes to fields. And in this particular panel discussion, it's one of the imperatives because we have to address increasing complexity of pest and disease pressure. And we also need to discuss about the various strategies in terms of rapid or quick development of better breeding lines, which can tackle multiple biotic stresses, and how we can integrate conventional breeding with molecular tools, AA, and also phenomic tools, and how to bridge the gap between research, varietal release, and then farmers' adoption. There are a few questions I would like to ask the panelists. I'll just throw a few questions. We can have about a 3-minute submission by each panelist, following which we'll have a short round of discussion with the audience also. So the broad questions which the panelists can answer in addition to telling their own views is that, my first question is, are we over-relying on major genes? And is there any scope or limitation of deploying the minor genes or QTLs in a breeding program? How do we predict resistance breakdown before it actually happens? Are there any strategies? And the third question is, can artificial intelligence and machine learning reduce breeding cycle time meaningfully? The fourth question is, what is the ideal gene combination particularly for Indian ecosystems? Are we there? For example, for bacterial blight, I can say with confidence. And similarly, based on the presentation given by Dr. Srinivas Prasad and Dr. T.R. Sharma, we are also there with regard to blast resistance. But with regard to the other insect pests and diseases in different crops, can we tell what is the ideal gene combination depending on what particular ecosystem in which you are growing? And my final question is, how do we ensure pyramided genes, how can they remain effective post-release, or how to prolong the life of the pyramided gene products? These are all broad questions which the panelists can attend, but I would like each one of them to submit their views or and try to answer at least one of these questions. I now invite from my right side, Dr. Viresh Godaji. You have about 3 minutes to give
Sure.
submission.
Thank you, Dr. Sundaram. So my viewpoints, especially right kind of a gene combination or right approach to tackle some of these challenges is it's always what customer needs. First, we need to think from the farmer what farmer is actually facing a problem. And it should be not across India or across overall rice community we want to solve. It is always essential to know that what product and what product concepts you wanted to build. Based on that, I think if we can really design a right gene combinations, looking at the challenges what customers are facing, maybe that will help us to address some of the challenges. On the top, yield is always important. Every time, without yield, any upgradation we do on the particular product, it will be not completely accepted by customer, because customer always or a farmer always wants improvement over what he is doing today. Not only just addressing the challenges, it is also essential to have yield improvement. That's where I can say a lot of now new technologies which are available, improving yield along with bringing a lot of this the tolerance. It will be a right approach, I can say. And most of the genomics what we learned today, if you can look at the animal genomics, I think a lot of advancement happened even before the plant. So maybe it's a good approach to look at what advancements are happening in animal genomics, which will actually help us to bring a lot of quantitative approach to tackle a lot of this major and minor genes what we are looking at. So always we need to keep a open, high, and thinking process to look at some of the advancements so that we can really tackle site-specific or target-specific problems. We can bring it for the customers.
Thank you very much, sir. I now request Dr. Devaraja Achar from Indo-American Hybrid Seeds to give his perspective on the topic.
So I would be discussing on marker-assisted breeding. That is one of the important key aspects for any kind of a trait improvement or development of a new varieties, in fact. As Sundaram sir said, from car to SUV, in fact, it was if you see the advent of the markers in almost 22 decades, majority of the varieties have been, mega varieties have been developed and have been commercially, viable in the market. So therefore, identification of key traits is very important. But if you see the complexity of the trait and the evolution of these races, in fact, with respect to the fungal and bacterial races compared to the virus, in fact. As we are pyramiding the time taken, what we are taking the pyramiding the genes or something like that, it is tenfold higher than what the strains or the pathogens are evolving, in fact. To combat and cope up with such kind of things, it's very difficult, in fact. So that's the reason where the gene editing approach is one of the new approach where you can able to do it within short of 2 years, where you can anticipate, in fact. And now, not the developing or incorporating the resistant gene to the background. Now you are going to edit the same plant by recessive ways. That means either you are making the plant to be not recessive or resistant to for a particular disease, in fact. So therefore, the fast tracking of those incorporation or editing the gene of the native genome is one of the best approach than putting the donor gene into this, donor genetics into this one, because of the lot of linkage drag and that and this. Okay. Of course, there are many approaches have come, genome association or other ways of these things. But pyramiding the genes in the native, in the major genes are fine. But if you take the QTL, again, it is a complex with the genome editing. So therefore, a lot of issues are there, but as a lot of marker-assisted breeding is need to be fast-tracked with the multiple tools, in fact. The best approach is presently the gene editing approach, which we need to take it forward for the varietal development or hybrid development in rice. Thank you.
Thank you very much. Now I request Dr. Venkatesh Kulkarni, Director R&D, Nath Biogene Seeds, to give his perspective.
Good afternoon, everybody. I am actually placed between two people who both are helping us in, especially rice marker marker-assisted selection in rice. Whenever my laboratory is overflowed, Dr. Devraj Acharya helps me in analyzing our sample and sending the results to us. The way in which the cost and the speed that he manages is very, very appreciable. He has helped us a lot in developing our population, etc. The second one is Dr. Sundaram, because in my first part of rice marker-assisted selection, honestly, we suffered a lot because of the source. The markers were in such a source that at the end of the day, product was not coming. Now, I am very happy to say that the elite sources are being impregnated with the markers, and those are available for as a source for much more validated sources are available for development of especially blight and blast. I have some of the points that to be successful in these marker-assisted selection are the hotspots and as well as screening protocols. And a source of the material is very important. These three things, a consistent management of hotspots and pure culture of the organism will help us in getting the fastest and accurate results. In addition to that, your period of marker analysis and before flowering, if the marker data is available, then we can really manipulate within the field and get the things. When we said that we are going for 4 markers, then we means 1 out of 16 will be our homozygous plant. So based on that, we increased the population and we got the results. That actually helped us. On and whole, in rice, it is working very well for marker-assisted selection to address the biotic stresses. And in addition to the rice, in tomato, we are seeing a lot of success on that. Here, one of the challenges as a private sector person, we are also working on hybrid breeding. So one of the protocols that we have developed is that once we develop a B line with the marker, then integrating it, making it an A line, we are reducing the time span, and we are doing some very good work on that. However, a lot of our platform is getting shifted to TGMS line. There will not be any big difficulties because it's as good as any pure line development. Durability question is consistent. Even we go through the marker-assisted selection. In the hotspot, consistent screening would help us in deriving a durable thing. Further, first few multiplication of breeder seed in the hotspot itself will also help in enhancing the durability of the material that we have developed. On AI, recently our team got, our R&D team got exposed to the AI from an US AI specialist. Our company is trying to make an AI agent for our R&D, and we are working on building the data. Historic data is very important for us. So I feel whatever I have learned till now and whatever I have experienced with a limited exposure to that, AI will have a lot of solutions to us with the quickest solutions from our historic data and future model building. AI can help us in a much better way in the coming days, because most of the agricultural science is built on a vast amount of data. For example, if you take your initial varietal trial, the amount of data that in that varietal trial is so humongous that it will be humanly impossible to assess the variation and means to judge the right variety as a superior. So I feel that AI will be a very great tool in the coming years for developing new models and identifying or decision making in breeding. With this, I thank US Dharwad and other organizations for giving me opportunity. Sir, thank you for me. I feel very proud to be sharing with you platform.
It's my pleasure also, sir. Now, sir, I now request Dr. Prabhugoda Patil, who is a senior rice breeder in Advanta Seeds, to give his own perspective. Okay, sorry.
He's
okay.
not here.
Okay, go ahead, sir.
So I'm Chandrasekhar from Kotiva.
Okay.
So,
okay, sorry, Dr. Chandrasekhar from Kotiva.
It's okay, sir.
Okay.
No. So I think we are like fortunate. Again, as I said, like I am also alumni of this college. Coming back here and talking in front of, especially students, is very encouraging. So from morning we saw like multiple presentations about the disease emerging situation and the new tools, how we can use those to effectively monitor, breed for the resistance, and the new question is about durability. I'll just try to address each one of those things. Like one, most of the time we discussed about deploying this major effect QTLs. I think which is progressed very rapidly, and especially on rice. I always think like in the insect world, Drosophila, and in the plants world, we always refer to Arabidopsis. But on a cultivated species, rice is having so much information generated in like multiple million base GBs, you can say. Like that much data is available on genomics, phenomics, everything is there. How to put that data into effective use and make meaningful decisions for breeding is actually a challenge. If you that much data is available, that is where these new tools, AIs and all those things will be helpful to make that right candidates. Because as a plant breeding students, we know that, right, the simple genetic gain what we make is time and cost. Making those decisions in time and putting it in the cost effective manner is sometimes we always think that it is only private, but it's happening everywhere because resources are infinite. So we need to make those decisions in right way. So marker assisted backbreeds, as I said, like multiples we have seen. One of the area which we have significantly developed and we are seeing a benefit of that is, especially in a self-pollinated crop like rice, genomic selection. Because we are trying to make a self-pollinated crop into hybrid crop, and there are challenges, like for example, I want to evaluate 10,000 test crosses. It's very difficult to generate those many test crosses, put those in field and evaluate. So that is where your genomic selection tools will help significantly and make a lot of predictions and generate thousands of recombinants and advanced right recombinants. Otherwise, it would have been not possible without these genomic tools. And another thing which we want to discuss about pyramiding. So in pyramiding is like, again, like you will see in rice, as I said, like significant information is available. You will have multiple genes reported. We talk more of like product, market, customer. Our language will be slightly different from a private perspective, but for everybody it is same. Farmer is a customer. So you can load multiple genes into everything, but you need to have that right strategy, where what works, and put that right combination so that you have to balance the performance for other yield things as well as the disease. So putting that combination is very important from that perspective, so that you don't have a yield drag, you don't compromise on the yield plus the tolerance. So another thing, just touch about our surveillance. Surveillance is one is human, the way we get our physical reports of this is reported there, that is reported there, but that gives more of a disease. But to go to the next level, biotypes, races, one of the approach which is evolving is molecular diagnostics. Like the way you characterize your the pipeline of the genotypes, you also characterize the pathogens for their genetic diversity, how they are emerging, and keep testing on your a set of lines, which we typically call as differentials. That is one thing which we can keep you ahead of the cycle and look for those breakups and what is that which is working. So in the climate change era, these breakdowns will happen even faster. So these are my perspectives, sir. Thanks for the opportunity. Thank you.
Thank you very much, sir. Now let's move to Dr. N.G. Ramachandra, who is the lead breeder rice breeding program at Bioseeds Hyderabad. Sir,
Thank you. Thank you, sir. Thank you for this opportunity. I think I thank Dr. Sundaram and all the organizers for this opportunity. It was a great place to be there. I think basically, if you look at the rice, is one of the, I should say I'm fortunate to be in the rice because a lot of information is available. I think from total genome is available with us. So that's a great opportunity. And also if you look at the public sector, we have a lot of institutions here in India, especially working in Hyderabad, so which are really cooperative and helping us in many of the things. Like we start with the screening. I think screening facility is wonderful in IRR, so which we can utilize. I think we are utilizing it presently and doing the things screening. Art spot screenings are one of the other things. I think these two are the artificial screening and the protocols available in most of the diseases like BLB, BLAST, BPH and all. So and also the upcoming diseases like false smart and also the viruses I think is developing fast. So but at presently most of the companies and most of us including our company, we are working on basically BLB, BPH and BLAST. So here the main thing is we have to look at in the farmer perspectives, what is required for the geography and what is required for the business. So if we load too many genes in one of the lines, so we are going to lose some of the yield genes. So yield is the main things. So first, for private company, yield is the one thing we should talk. So other additional parameters like health, hygiene factors are like BLB, BPH are the secondary there. But if we only concentrate the hygiene factors, leaving the yield is not good. So yesterday, I think some person asked a very good question. I think Dr. Krishna Kumar has answered him actually. So if you put everything in one line, so it may be super duper very good. It is doing well in all the gene factors, but it's going to lose the yield. So apart from the yield, I think rice is quality is also one of the important, quality and HRR. So if we don't lose, we have to maintain a yield, quality and HRR. So apart from that, if you have one or two genes which can complement the yield, and where adverse conditions, especially when the high breakout of BLB, BPH it comes. So that will be complementary for the things. So which is very important. But if you look at the developments in rice, I think most of the companies, most of the institutions are routinely using the molecular tools. So that is the best part of the rice breeding at present. So if you look at any of the companies, even the smaller companies which are using the molecular tools for the disease resistance, they are outsourcing the molecular work and outsourcing the screening facilities, they are doing well. So